Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Aina Shahila binti Bramin and my student ID number is 2024245056. Now I am going to tell you the overview of maltose. What is maltose? Maltose is a disaccharide sugar made of two glucose molecules that link together. Where it comes from? It is produced when starch is broken down, for example during digestion and malting grain. How it tastes or used? It's a sweet sugar used in brewing, fermentation, and food production. And how it digests? In the body, the enzyme maltase split the maltose into two glucose units for energy. Now, I am going to tell you the importance of maltose. Maltose is a readily available energy source for plants and animals. Maltose also important in certain industries such as industry and research, health consideration, and biology and nutrition. Industry and research, maltose plays a major role in brewing because it is the main fermentable sugar used by yeast during beer production, and it is derived from malted grains. It is also used as a sweetener in food products and as an inactive ingredient in some medicine. From a biology and nutrition perspective, maltose as an energy source providing quick energy for cells in plants and animals. It is easily digested and converted into glucose for absorption. However, from health perspective, maltose has a high glycemic index, which can cause rapid blood sugar spikes. Therefore, moderation is important to reduce the risk of health issues such as obesity and diabetes. Chemical information of maltose. Maltose has the chemical formula C12H22O11 and it is classified as a disaccharide. It is made up of two glucose molecules that are linked together by an alpha-1,4-glycosidic acid. The elements present in maltose are carbon, hydrogen, and oxygen. Maltose is formed by a condensation reaction where one water molecule is released during the bonding of the two glucose units. Pictures down below is the structures of maltose.
Assalamualaikum and hello everyone. My name is Nur Shifa Masara binti Muhammad Nawi and my student ID is 2024230532. Now, let's talk about the properties of maltose. If you've ever chewed on a piece of a bread for a long time and then you notice that the taste is getting sweeter, that's when you make maltose. Physically, maltose is a white crystalline solid. It is soluble in water and has a pleasant sweetness. But its sweetness is just as half as sweet as the sugar that you put in your coffee in the morning. Chemically, maltose is a double sugar or what we call it as a disaccharide. It is built from two glucose molecules that held together by what we call an alpha-glycosidic bond. Now, here's a key distinction. Unlike sucrose, maltose is a reducing sugar. This means it has a free end that can react with another chemicals which makes it really active in biological chemistry. Now, why does maltose really matters to you? It is all about the breakdown of energy. When you eat light starchy food like potatoes or rice, your body can't use that giant starch change all at once. Your saliva and your pancreas release enzymes that called amylase that chop that starch into smaller pieces. That pieces are maltose. Eventually, enzymes in your gut called maltase SNPs that maltose right down the middle, giving you two clean units of glucose. That glucose is the fuel that enters your cells to produce ATP, the currency of life. So, While maltose isn't the long-term storage, it is vital intermediate bridge that gets energy from your food into your blood. You might also wonder if maltose with things like cellulose with trees. The answer is not directly. Maltose isn't a structural brick. Instead, think of it as a metabolic signal. In the plant world, this is actually quite cool. At night, when plants can perform photosynthesis, they break down their stored starch into maltose. The presence of this maltose acts like a biological clock signal, telling the plant how to manage its energy reserve so it doesn't starve before the sun comes up.
As you know, maltose is a disaccharide that made up of two glucose molecules. Each glucose molecule has a hydroxyl group. So during the synthesis, one glucose molecule will lose hydroxyl group while the other one will lose hydrogen group. So the loose product will combine and form water, and the water will be released. The two glucose units are then joined by an alpha-1,4 glycosidic bond, and they will form the product, which is maltose. As you can see in this slide, the synthesis of maltose involves two things, which is condensation reaction and also formation of glycosidic bond. So the condensation reaction is like how I explained on the previous slide, while the formation of glycosidic bond has an addition information, which is the glycosidic bond is covalent bond that holds two monosaccharides together, which means the glucose is monosaccharide and two molecule of glucose, which is two molecule of monosaccharide, will form disaccharide, which is maltose, by the bond that holds them together, which is glycosidic bond.
Assalamualaikum warahmatullahi wabarakatuh. So, my name is Ahmad Shafiq Rasidi. My student ID is 2024410486. So, today I will presenting about current research and innovation of maltose that is disaccharide of carbohydrate. So for the first study and the research toward this maltose that is disaccharide, that is expansion of maltose related to transporter in ASCOM site and their association with corresponding disaccharide utilization. So this study is explore how different type of fungi absorb sugar using special gatekeeper that is protein called transporter. By analyzing the DNA of a wide variety of fungal species, research discovered that not all fungi eat sugar the same way. One specific group known as Comcota, which is which include yeast and mold, was found to be particularly efficient at consuming maltose. The study revealed that these fungi have evolved to possess both more gates to let this sugar in and more internal tools to break them down. Understanding this genetic setup is valuable because it allows scientists to engineer better fungi for industrial use, such as efficiently converting plant sugar into useful product. So for the second innovation of this sugar, that is trichotyl carboxymethyl cellulose and polyvinyl pyrrolidine and maltose encapsulated onto solid microneedles to increase the transmitter drug delivery. So, this delivering medicine through the skin is a great alternative to pill or injection, but the skin outer layer is a tough barrier to cross. To solve this problem, scientists are using microneedles, tiny needles that are painless to penetrate the skin to create a pathway for the medicine. So researchers showed these microneedles are effective for delivering various treatments, including insulin and vaccine, without hurting a patient. This review explored the different type of microneedles, how they work, and why they are promising technology for the future of medicine. So by looking at the diagram given, so we can see that there are many microneedles. So this microneedle will injected into our skin. So the function of the maltose coated at the microneedles is functioned to increase the transmitter drug delivery to our skin. For the third innovation in maltose, that is self-assembly of maltose albumin nanoparticle for efficient targeting delivery and therapy in liver cancer. So, scientists have developed a new smart delivery system to send chemotherapy drugs directly into cancer cells using a tiny particle made of sugar, that is maltose, and protein. They loaded this particle with a drug, that is doxorubicin, and design them to release the medicine only when they are detect a specific chemical environment inside the tumor, like low pH. So, because of this, cancer cells are hungry for the sugar, they quickly absorb this particle, which allow the drug to enter and kill the tumor cell effectively while sparing healthy cells. Tests show that this method significantly stop the tumor growth and reduce the harmful side effects usually caused by chemotherapy. For the fourth innovation or research in this disaccharide, that is glycoconjugated vaccine that containing Escherichia coli O157H7O antigen linked with maltose binding protein elicit humoral and cellular responses. Okay, scientists are create a new vaccine to fight the dangerous E. coli bacteria by attaching a specific bacterial sugar to a carrier protein called maltose binding protein (MBP). While previous research mostly focused on antibody production, this study discovered that the new vaccine trigger a powerful dual defenses system in mice. It successfully produced antibodies in both the blood and the intestine of the neutralize the bacteria, while simultaneously activating T cell cellular immunity to directly attack the infection. The result proved that using MBP as a carrier is highly effective because it stimulates the immune system to fight the pathogen from the multiple angle. Lastly, for the current research of this maltose sugar disaccharide, there is the generation of a novel Saccharomyces cerevisiae strain that exhibit the strong maltose alternation and hyperosmotic resistance using non-recombinant technique. So, in this study, maltose function as an essential fuel such for yeast when making bread that has no added sugar, nor as unsugared dough. To make a bread rise, the yeast must be very efficient to act absorbing and digesting this sugar, which require high level or specific internal tools called maltose permease and maltose. These research found that yeast strain possessing these active tools are non-lagging, meaning that can start fermenting and producing gas immediately without delay. Consequently, the ability of to analyze maltose quickly was used as a key strain that performed a better in baking. So, as we can see, the difference in maltase and maltose permease, that is, maltose permease is producing more gas or strangle in the table more than Maltese. So this result show that Maltese premise is more suitable to use in this research more than Maltese. That's all for me. Thank you.
Next, there are several applications of maltose. Firstly, maltose is used in brewing and fermentation process. Maltose also function as carbohydrates in animal feed and support microbial activity in soil. It is also used in oral medicines because it provides a stable energy. Maltose serves as a carbon source for microbial growth in fermentation. It is also used for development of biomaterial like biodegradable polymers. Maltose play a role in nanotechnology as surface modifying biofuels as an intermediate sugar in fermentation. And lastly, it is used to help reduce dependence on petroleum-based chemicals in green chemistry. Implication of maltose is it provides a rapid source of energy due to its easy digestion and conversion into glucose. However, it lacks its essential vitamins, minerals, and fiber, making it a source of empty calories. The health benefits of maltose is it allows for rapid absorption and utilization by the body. Maltose also has a health risk because it has high glycemic index, excessive consumption, and can lead to rapid increases in blood glucose level. This may contribute to insulin resistant weight gain and dental problems when consumed frequently.
So we are at the end of our video discussion. So this is our reference that we use in this study and in this discussion. That's all from us. Thank you.